Good afternoon. I'm Marcia Bay-Childress from the Center for Biomedical Ethics and Humanities, and I'm happy to welcome you to today's Medical Center Hour, a program entitled Retroviruses, Reproduction, and Regenerative Medicine, the Influence of Federal Funding. We're pleased today to welcome Anderson Kiessling from the Harvard Medical School and the Bedford Stem Cell Research Foundation, and also importantly, an alum of the School of Nursing. And we have, as part of our introduction today, the introduction of our speaker and presentation of an award. First, I'd like to welcome the Dean of the School of Nursing, Dorrie Fontaine, and we'll proceed with the award and then with Anne's talk and then your discussion with her. Thank you. Welcome, Dorrie.
Thank you. I'm pleased to welcome everyone to this very special lecture, and I want to thank the Medical Center Hour for their sponsorship with us and collaboration on today's program. We've had some wonderful collaborations this past year, and I want to very much thank you, Marcia. So in nursing as in medicine, we have many remarkable alumni making a difference in the lives of others, and the nursing alumni that you're going to hear from today is certainly one of those. I like to say you never know where a nurse is going to turn up, and that's today. Anne Anderson Kiessling received her Bachelor's of Science in Nursing from UVA in 1964, and then went on to get a BS in Chemistry and an MS in Organic Chemistry from Central Washington University, followed by her Ph.D. in Biochemistry from Oregon State University. During the 70s, her postdoctoral research explored relationships between viruses and cancer and led to the controversial discovery of reverse transcriptase in normal human cells. Believing that reverse transcription may play a role in early embryo development, she began to study eggs, sperm, and early cleaving embryos. Her expertise in reverse transcriptase in the reproductive tract led to the first studies of semen transmission of the AIDS virus in 1983, and in 1985, Harvard recruited her as associate professor in obstetrics, gynecology, and reproductive biology. She is currently Associate Professor of Surgery at Harvard Medical School. Thus my comment, you just never know where a nurse, especially from the University of Virginia, is going to turn up. In the 1990s, Anne combined her background in human egg biology and AIDS with her interest in stem cell research. Given the status of federal funding for this controversial type of clinical research, the topic she's going to speak about today, she founded the Bedford Stem Cell Research Foundation, and she will talk to you about, you never know when you're going to wake up and be a foundation director. At Bedford, Anne developed the first program of assisted reproduction for couples living with HIV disease, and the first human egg donor program for stem cell research. She's the author of the first textbook on this very controversial topic, and she's published countless scientific papers and lectured internationally on the topic. Under Anne's leadership, the Bedford Stem Cell Research Foundation has developed promising advances in deriving patient-specific stem cells while also tackling the moral and ethical issues in stem cell use, issues that I know you're all very interested in. Its programs have helped more than 120 couples affected by HIV disease have safe, healthy babies, and one day may lead to cures for spinal cord injury, Parkinson's, and HIV disease. So for all these reasons and more, the School of Nursing Alumni Association last summer named Anne the very first recipient of a new award they had just established called the Alumni Achievement Award. So please join me in presenting this award. To join me in presenting this award will be my wonderful partner, the head of the Nursing Alumni Association and the class of 1969 at University of Virginia, Miss Patricia Woodard. So Pat.
Thank you, Dorrie. First, I would like to thank the members of the awards committee who participated in the selection process. The committee included Julie Grisha, Suzanne Furmeister, Sarah Delgado, Sarah Kirshner, and Catherine Reed, and was chaired by David Strider, and we thank each one of them. And I'm not sure whether you know how that you were one of the inspirations in the creation of this award. This new award honors an alumna or an alumnus of the School of Nursing who has gone on to show superior achievement in a field other than nursing. It recognizes distinction in one's field and/or contributions to another profession, to business or industry, to government or public service, education, science or technology, or for service to humanity. As nursing students are so often told, once you're a nurse, you're always a nurse. But we know that the knowledge and skills derived from a UVA nursing degree may lead someone to many other amazing accomplishments in life as well. And you've certainly proven this to be true. Ladies and gentlemen, please join me in congratulating the School of Nursing's 2010 and first ever Alumni Achievement Award recipient, and our speaker today, Dr. Anderson Kiessling. Congratulations.
Thank you. Please leave us. Thank you. Wow. Thank you very much. It's a little hard to follow that. There's a couple of things I wanted to add to what was said today. I've actually never considered myself leaving the field of nursing. A lot of what I'm going to tell you today has a lot to do with human subjects research, and I've always felt it was a lot easier for me to organize a human subjects research, even the consent form, because I knew what people needed to know, I knew what patients wanted to know about what was going on. So I don't think I've ever really felt as though I left the field. And the other thing I want to add, partly for the women in the student, if there's any women students in the audience, is that I'm also the very happy mother of 4 wonderful children and 2 grandchildren. So I don't want you to think that you have to give up that part of your life if you want to go ahead and do sort of some independent work. Now I spoke with Marshad, I put together some science, but now I've taken back some of the science because I understand this is more of an ethics forum. And I've actually written on that topic, and I have developed some passions in that area. And I've put together quite a bit to tell you today. It might be easier to see the slides if some of these front lights were out, although I certainly don't want anybody to go to sleep. Is it okay? Can you see?
She'll see if the technician's.
We understand that John is on vacation. So I'm going to start with something that started at the very beginning of my career, and I think makes an interesting story for the role of federal funding in basic biomedical research in the U.S. And I think this is something that we now have to re-examine, and I'm going to give you, the whole talk today is going through some of my own personal experience with the idea of we're going to have to come up with a new paradigm about how to do this. So now this begins with very ancient biology that everybody in this room knows. There's a central dogma that was developed around in the 50s that we know now that our genetic information is made up of DNA. That genetic information is transcribed into RNA, and that RNA creates all the proteins that are everything we are. This has been the central dogma. This was challenged, and it's the challenge to this that I think is the beginning of the federal funding issue. Exactly 100 years ago, just after viruses were beginning to be understood, an agent was isolated from a tumor in a chicken that was called a filterable agent, and it caused cancer. And this was the very first time that cancer had been ascribed to an infectious agent. It was a landmark discovery. It was exactly 1911. And it was a retrovirus. So 100 years ago, we discovered the first retrovirus. And of course, HIV disease is a member of that family of retroviruses. And the first thing we knew about these viruses was they caused cancer. Now, 50 years later, a young scientist discovered that there was something about this virus that challenged the central dogma. And can you guys see this slide okay? Okay. And what he discovered was that although this retrovirus had RNA as its genetic information, so it had an RNA genome, what we knew about viruses at this time was that if they had an RNA genome, like poliovirus, they multiplied themselves by making another copy of their RNA and then making all the proteins they needed. This virus was different. And Howard found out that it was an RNA virus, but it required DNA synthesis to make copies of itself. Absolutely nobody believed him. And this paper was published in 1964, which I think is interesting since that's the year I graduated from school here. And of course I wasn't paying a lot of attention to this field then. But this paper was published in 1964, and it took him about 4 or 5 years to get this work published. He presented it at meetings, he was essentially booed. And what he discovered, by 1971, others had repeated his work, and now it was clear that this virus had changed the way we understand biology forever. This is now 50 years ago. So you do indeed, you go from Rous Sarcoma Virus RNA to a DNA copy of that virus, back to new RNA for progeny virus, protein to virus. So this was reverse transcription. Now, interestingly enough, when Howard discovered this, he thought, this isn't a virus pathway. This plays some fundamental role in biology that we don't understand. This whole way to do this has got to be not something that a virus invented, but it's something that a virus has co-opted to increase its productivity. But his work, I think, is a really good example of overdependence of the power if we're totally dependent upon federal funding, because Howard could not get his work funded. His applications to the National Institutes of Health were always turned down for about a decade. The only reason he got this work done was because the University of Wisconsin had a McCardle laboratory that studied cancer. And because the University of Wisconsin had faith in his work, they felt that the work was sound, they funded him. And that's how he was able to do his Nobel Prize winning work, for which he got the Nobel Prize in 1971. Now, when he found this whole process, there's an enzyme involved called reverse transcriptase. And he decided that this gave a really interesting opportunity to essentially amplify genes without changing the Mendelian inheritance of genetic information. So for instance, frogs do this. When frog embryos are developing, they all of a sudden amplify the ribosomal gene so that you have thousands of copies because you need to make ribosomes really fast during early development. This would give an organism time, and we don't know exactly, we still don't know how this works, but it would give an organism an opportunity to amplify copies of genes it needed at a definite stage of development without changing what was inherited. And this he called his protovirus hypothesis. Now, this was when I was just finishing my graduate work now. And I had done my thesis work, my Ph.D. thesis work on leukemia. And I think I developed a real interest in cancer and leukemia while I was in nursing school. And I studied the virus that caused leukemia in chickens. So it was a similar family. It was a retrovirus that caused cancer. And I was very interested in the concept that this reverse transcription might have a normal role in biology. If his theory were true, it would predict that you would find reverse transcriptase activity in embryonic stem cells and perhaps in cancer. It would also predict that you would find copies of transcribed genes in embryonic cells and in cancer. So we set about, for my first postdoctoral experience, we set about trying to find reverse transcriptase activity in normal human cells, and we did. And it took us 2 years to get this work published. This was very unpopular because at a time in the early 70s, there was clear vision on the part of many people that the reverse transcriptase was present only in tumor viruses, and that if you found reverse transcriptase in malignant cells, it was because they were infected by a human tumor virus that caused leukemia. This was very entrenched at the National Institutes of Health. and some very major National Institutes of Health scientists kept this work from being published for a couple of years until they got the new congressional funding. So this work was not allowed to be published until a new NIH budget had been approved, which was part of the war on cancer that had been introduced by President, what was his name? Nixon. So in this work, we discovered actually that reverse transcriptase activity was present in every cell we looked at. Some cells it was higher, some cells it was lower. But if you were careful, you could find it everywhere. And it wasn't that much higher in the cancer cells. So this led me then to think about, okay, the next thing to ask then is, can you find reverse transcriptase activity in early embryos? That's the next question. And I happened to be lucky enough to I went to Oregon Health Sciences University as a faculty person there. And we actually did find, at the time reverse transcriptase had a number of names, and this was one of them. And we found reverse transcriptase activity in early mouse embryos. And Dr. Whiteloff taught me which end of a mouse to get eggs out of, and which end of a mouse to get sperm out of. One of the things I had done as a graduate student was vow I would never do an experiment with mice, and I would never do an experiment with tissue culture. So of course here I was doing both. So we did find reverse transcriptase activity in early mouse embryos, and we also found that if you cultured those embryos in the reverse transcriptase inhibitor, AZT, that the embryos failed to develop. But that was it. The tool, we could just barely had enough tools to even ask this question. This is 30 years ago. This work was done 30 years ago, and this is as far as we could get right then. But interestingly, and I put this slide in for Dr. Hinton, who's in our audience, interestingly, in the course of doing this work, we discovered that there was an incredible amount of reverse transcriptase activity in a very interesting organ in the male mouse called the epididymis. So this is a testis. This is the epididymis of a mouse. And this organ has a number of really interesting features, one of which is very unusual epithelial cells that have stereocilia at the tips of it. We don't know a lot about this organ. There are a few scientists in the world who study the epididymis, and Dr. Hinton tells me that we're not all getting as old as I thought we were, because it seemed to me at these meetings everybody's getting really old. One of the things about this organ that's so interesting is it doesn't get cancer. And that flies in the face of the level of retrovirus activity we found in it. Now that we've sequenced the human genome, now we know that 40% of our genetic information is retroelements. 40%. Now, retroviruses are not the only type of retroelement. We have a number of truncated viruses that will take up a whole week's discussion on its own. But 40% of our genetic information is retroelement. Now, part of what we see here, what we published, these are all 25 years ago now, was that we found an incredible amount of reverse transcriptase activity in the fluid from the epididymis of these male mice. It was an astounding finding. And at the same time, when we did electron microscopy of it, you can see the particles. So these are all virus particles. These virus particles are being produced by retroviruses that are part of the genetic information of this mouse. These are not infectious particles. They're part of the mouse's genome. And for some reason, they're turned on in the epididymis specifically. Now, they might also be turned on in the spleen. That was hard to nail down. But this was an astounding finding that this retrovirus, which is part of this animal's genetic information, is expressed at really high levels. Here's some more. Each of these particles, this is a cross-section through a sperm tail. This is a cross-section through a sperm head. And you can see these particles are lined up against the head and the tail. This was an extraordinary discovery. Nobody knew what to do with this, including us. And this work actually hasn't been funded for a while. But recently, there's some new tools available. And this was an interesting paper that was published in 2004 by a group that has now been able to show that there is ongoing active reinfection of human genetic information by retroviruses that are part of our genetic information. So there's something going on in the reproductive tract that allows these viruses to be expressed, and they get reintegrated into new sites at fertilization. This is not something we understand. We don't know if it has a role in disease. It obviously has a role in evolution. And a few years ago, we were actually able to return to this work a little bit ourselves. And in 2007, we were able to use some new genetic tools, and we were able to look for 6 classes of what are called human endogenous retroviruses, HERV. And this is type K, type 9, type W, type E, type R, type L. And we were able to show that, or we found that the epididymis expresses all 6 of these classes of endogenous human retroviruses as RNA. We have not seen these by electron microscopy. We do not know if they make whole virus particles, but they express the RNA, whereas the testis only expresses a few of those. Very interesting finding. What about infectious retroviruses? So what about people that are infected with HIV disease? Is this virus become part of, can it infect cells in the epididymis and get turned on? So in 1983, I just want to remind this group, before many of you were born, 2 things happened that changed medicine or changed the world forever. And one was that HIV disease, or AIDS, was recognized as a sexually transmitted disease. That was also an astounding discovery. And the very same year, in vitro fertilization, or IVF, was recognized as standard of care for infertility in the US. It had been ongoing in England for a few years before that. So at that point, I was still in Oregon, and because my lab had been studying retroviruses in the male tract, we did the very first studies just to see if we could detect HIV disease in human semen. We'd never seen a human sperm before. We detected millions of particles in the semen. And we were asked, because we knew how to fertilize a mouse egg, we were asked if we would help set up the first IVF laboratory at the Health Sciences Center. And so that charted a course for the rest of my research career, where we're doing these parallel tracks. We're trying to understand how HIV gets into semen, because in theory it can't. And we've been trying to understand, is there a role for this in early development? Now this is an old electron micrograph this is an HIV particle right next to the cross-section of a mid-piece of a human sperm. We know that there's HIV in semen. We've studied this now for many years. And I'm just going to summarize what we know. The big question is how does HIV get into semen? And the answer to that is that we still don't know. Now, the male tract, and I won't go through this in detail, but the male tract is basically designed to protect sperm from the immune system. Little boys don't make sperm until puberty. Your immune system responds to sperm as if they were bacteria. And so the whole purpose of the male tract organs, so if it's protected from the immune system, how can a virus that infects the immune system get into semen? And we can talk, if anybody has specific questions about that, I can return to that. But what we do know is that the HIV burden in blood and in semen is very discordant. These are 2 compartments. The genetics of the virus in blood and in semen is very discordant. And so we know that there's now somehow, especially for people who go on antiviral therapy, these become 2 different compartments of virus infection. Now in 1989, and this was way too soon for the health of my career, I wrote a little letter to an editor that said, of a fertility journal, that said somebody was going to have to help figure out how to help people living with HIV disease safely parent without passing on the virus. In particular, we were concerned about the 10,000 men and boys with hemophilia that had been infected with blood clotting factors. How were they going to have their families? Because it was clear that very few people die of HIV disease acutely. It takes a long time to get sick enough to die of this disease. And antivirals were being developed at a great rate. So it was very clear that there were going to be a whole population of people HIV infected were going to be around at least and want to parent. It was too soon to talk about this because it was too scary to think about somebody with HIV disease having children. But as soon as this article, so this was a little hard, it was a political problem for me for a number at work. But because we published this, the phone began to ring. People who were infected began to call and say, what are you going to do about this? And so this again is another example of being overly dependent. We Our National Institutes of Health has never, ever been able to fund research on fertilized human eggs or early embryos. And I'm actually going to, at the end of this, I'm going to tell you why. So we've never been able to do that. Consequently, in contrast to most clinical treatments, assisted reproduction does not benefit from basic research, basic federally funded research, either for the mom or for the offspring. Until recently, programs of assisted reproduction had not been creating a research fund to do this. About 3 or 4 years ago they started. But for many years, every time I got to talk to a group of assisted reproduction clinicians, I reminded them that the work wasn't funded because they weren't creating a research fund. I was frequently not invited back. Consequently, after 3 decades of creating tested babies, there is still no test to let anybody understand which embryo in that dish is viable. You're looking at 10 embryos, and there's no way to know which one the mother should get back. Consequently, too many are transferred to ensure pregnancy, which has led to the epidemic of twins and triplets. This is an absolute travesty. And it's only partly, I think, the fault of the federal government. Our federal government has strong reasons for not wanting to fund this research. Somebody else should have funded it. Finally, in 2007, after being criticized for a long time for this whole program that we developed to how to help people with HIV disease, parent was given a prize. The prize was actually awarded to us by somebody who'd been one of our biggest critics, which is the way that these things have a way of coming around, right? This is just a quick summary of the program. I won't go into the details. The patients began in 1996. It was very controversial, and it was caught in a hospital merger, so that's how I woke up one day a foundation director. A Massachusetts public charity was created, and we even at the end had to create a mobile clinic because we were searching for a permanent clinic. That's a picture of it. And it was actually a great, clean little clinic. And the first baby was conceived in 1998. An NPR snoop found out about this program and came, and his name was Stephen Smith. And this is an interesting, it's posted on NPR. It's a great story. He won some kind of a prize for this. And I spent weeks begging him not to come, but he did. It's an interesting story. So this is the first baby. This is a dad with hemophilia. We've actually lost him. And this is now first baby as a baseball star at age 10. And as of this month, the lab just tells me that the program has 46 collaborating fertility clinics all around the country now. This is now something that's routine. There have been 146 babies born with 11 ongoing pregnancies, and all the moms and babies test negative for HIV. I assumed that when we got this organized, fertility clinics would pick this up and carry this ball, but it turns out they haven't. So we're still doing this. Now let's switch quickly to stem cells, because this is the other thing that the foundation has gotten caught up in. This is the cover of Science in 1999. So 12 years ago, we were on a roll about stem cells. How did this come about? And this again is the third example of an overdependence on federal funding. So because of the moratorium on federal funding, human embryonic stem cells had to be privately funded. We had had mouse embryonic stem cells for 2 decades. And mouse embryonic stem cells have been an enormously effective tool in genetically engineering the genes in mice and understanding some early development functions. We also had some stem cells for monkeys, but this work couldn't be federally funded, and so everybody thought it couldn't be funded at all. Geron Corporation went from university to university asking if their investigators who were good at this could fund the work. Again, it was the University of Wisconsin who stood up to the plate, and they said, we'll take your funds. And of course, they had a scientist there who'd been practicing making stem cells from monkeys. So that helped too. And I'll go through that work. But let me remind you how this works. So this is a human egg ready for fertilization. Sperm enters. The egg begins to divide, goes to the morula stage, and becomes a blastocyst. Now, most of the people in this room have had this. And just to remind you that this ball of cells that's on the outside is going to become placenta. The little group of cells on the inside are going to give rise to the fetus. It's called the inner cell mass, or ICM. And that is the derivation of stem cells, right? So you take out this little mass, put them in a dish, and now we know how to culture them to become a line of stem cells. Oh, I added this little movie for those of you who've never seen an egg cleave. This is mouse. So this is a 2-cell embryo. This one's going to a 4-cell. You can see that early cleavage events, the cells get smaller and smaller and smaller. And it's only at about the inner cell mass stage that they become the size of a normal cell. So these early cells are huge. This is a human blastocyst. So this again, this outer cells here are going to become placenta and amnion. And this little knot of cells down here is going to become the fetus. And at about the stage of this, there's only about 40 or 50 cells here. So to derive a line of stem cells, you have to come up with conditions that allow those 40 or 50 cells to continue to develop in culture. So when it was first reported in 1998, Jamie Thompson, who was at the University of Wisconsin, funded by Geron, and he went to great lengths to set this lab up. It was kind of like overkill. He put it up across the street from the university, even bought separate lab coats so that there would be no conflict with his federal research grant. It was a little overkill and it was kind of high drama, but it was very effective. But the most important thing was that from 14 of those inner cell masses that I just showed you, he was able to derive 5 stem cell lines. Now, that in no way translates to 14 fertilized eggs. Only about 20–30% of fertilized human eggs go to that stage. But if you could get them to that stage, this efficiency of derivation was very promising. It's like 30% efficiency. And people began to do statistics, and they were able to show that if you look at all of the tissue matches that you need, so obviously regenerative medicine is going to depend on tissue matching like kidney donation and bone marrow transplant does. It looked as though you could come up with, if you came up with a bank of 200 human embryonic stem cell lines, you might be able to cover most of the tissue match needs for the population. So that was very exciting. That was 12 years ago. So immediately, President Clinton asked his National Bioethics Advisory Commission, chaired by Dr. Harold Shapiro, to look into this. And the commission came back with the following guidelines for federal funding. They thought that the derivation of embryonic stem cells from leftover fertility center should be allowed. This should be federally funded. They thought not eligible for federal funding should be the derivation of ES cells from IVF embryos created solely for research purposes. And everybody was sort of in favor of this. Nobody wanted people running around making embryos for research purposes. That was too hard to think about. And there was something called somatic cell nuclear transfer, which I'm not going to go into today because there's never been any stem cells derived from it. So at this point in time, pluripotent stem cells have been derived from two types. Oh, President Clinton didn't go along with those guidelines. They were recommended that we fund the derivation of stem cells from discarded embryos. President Clinton decided that was politically too charged, and I think the College of Cardinals helped him with that decision. And so that work was not fundable at that point in time. So no stem cell work was fundable at any time during the Clinton administration. Now, pluripotent stem cells have been derived from two types of blastocysts, not just embryos, but there's something called a parthenote, that if you don't learn anything else today, I'd like you to take away the concept of parthenogenesis and the value of parthenotes as stem cells. And parthenogenesis is similar to fertilization, except that there's no sperm involved. So it turns out that eggs can be activated in lots of ways, and they like to be activated. That's their mission. The mission is to activate and start dividing into smaller and smaller cells. Now the good news for guys is that if you do this, you don't ever give rise to a fetus. There are some ancient claims in medical literature that by women that this daughter that she has must have been a parthenote because there was no possible way that it could have been fertilized. But those claims have actually been pretty well disproven. So to date, there is no evidence that these parthenogenically activated eggs can give rise to a fetus. And so this seems to be a really reasonable, but they do give rise to stem cell lines. So there's something about the sperm that's required for appropriate organization of organs, but not necessarily lines that develop in culture. Now, the first primate parthenote stem cells were derived from monkeys, and this work was reported in 2002, almost 10 years ago. This is what an activated monkey egg looks like. I didn't show you any fertilized eggs, any fertilized human eggs, but they have two nuclei, one from the sperm and one from the egg. Obviously, a parthenote only has one nucleus because it only has egg chromosomes. And they begin to divide. They look like relatively normal early cleaving eggs. They go to blastocysts. Now, this is the egg donor. This egg donor is still alive and well. This work was done in collaboration with Wake Forest. They have a monkey colony that they used to study alcoholism. And they were interested in cloning. They have a group, evidently a troop of monkeys that have free access to alcohol all day long. And they divided themselves into teetotalers and into alcoholics. And they wanted to clone some of each of those. So they contacted Jose Sabelli, who was the author, the primary author on this work, because he was really good. He cloned cattle. And that was how they got involved in this work. And so I had these slides, and I had asked Jose many times, which was this monkey? Was she one of the teetotalers, or was she one of the alcoholics? And when I gave this talk in Tennessee once, and somebody in the audience was familiar with this colony, and they told me that she was one of the alcoholics. So we're wondering if that explains the smile. I don't know if you can see it. And I think today that this monkey may still be alive. But this is the inner cell mass from which the stem cell line was derived. And the efficiency was very similar to what was reported by Jamie Thompson's lab. So they started with 22 mature eggs. They're parthenogenically activated, and this is no sperm. They got 4 blastocysts, and out of those 4, they got 1 line of stem cells. Now those stem cells have been used for a bunch of studies now having to do with, there's a monkey model for Parkinson's disease. And these parthenote stem cell lines, it turns out, are relatively easily turned into the types of neurons that you need to treat Parkinson's disease. And there's a lot of ongoing studies with this particular cell line in the Parkinson's model in monkeys. Now let's turn to what's going on with human parthenotes. So this is a mature human egg. And early in the stem cell debate, it became the whole debate around whether you should ask women to donate eggs for research. Should women be asked to go through an egg donation cycle for research purposes? And the debate sort of took on its own, and it started in California when California organized its stem cell bill with a group of people there who wanted to make sure that women were not being taken advantage of for this new technology. And that's a debate that sort of has colored everything. Here's an article that we wrote about what are the advantages to parthenote stem cells. Nature asked us to do this. So obviously the advantages to parthenote stem cells is that they're not fertilized, so they're less controversial. They have half the tissue antigens as embryos. It isn't clear whether half is that useful because there's lots of tissue antigens that need to be matched, but they do have half. And they're obviously going to provide matched stem cells for ovulating women. So if you're an ovulating woman, say with spinal cord injury or diabetes, your eggs might be a source of stem cells for your own treatment that would be tissue matched. And again, a bank of a couple of hundred lines from these unfertilized eggs would match about 90% of people. So this is, I think, a very interesting resource that is underdeveloped, and it's underdeveloped primarily because of a quirk that doesn't allow any federal funding for this type of research. We just talked about that. Should women be asked? This is a huge topic. We tackled that because somebody came to us and said, please help us organize a stem cell program for women donating their eggs. And I won't go into all of that, but we had to organize a lot of ethics committees. We had this reviewed by a lot of people. And then one day we got brave and we put an ad in the Globe, the Boston Globe, that said, research team seeks women to donate eggs for stem cell research. And we waited for the phone to ring or the bombs to go off, and nothing happened. So then we decided maybe nobody reads the Boston Globe because reporters didn't even call us. So then we put the same ad in some local journals like Parenting Magazine and that sort of thing. One of the criteria that we had for egg donors, which I think is extremely important, was that they already be parents. We were not interested in young women who had not had their families donate for this kind of research. And I can go into that if somebody's interested, but all these women had to be parents. So yeah, there was a whole bunch that we've had to do a lot of advocacy with this. I won't go through all the steps, but it's a very comprehensive program. We were very proud of it. It's been written up in the Hastings Center. And it was also written up in Science when we did an abstract on it. And as you can see, by the time we got through with this, one cycle for these women was $25,000. We averaged about 10 eggs, so that every egg for research was going to be $2,500 an egg. You plan those experiments very carefully. It's a little bit like planning, some experiment that the marine biologists do. So the program is up and running for 4 years. This is a human parthenote. I don't know if you can see, it looks like the monkey parthenote, this one nice nucleus in the middle. This is what activated human eggs look like. They begin to cleave. They don't look nearly as nice as the primate eggs. And in fact, we never got a stem cell line. We then turned to frozen eggs because in the time we were learning to do this, the world also learned how to cryopreserve human eggs, which was not possible in 2000, but it's now very possible. And again, the frozen eggs went through the similar, you begin to get stem cell lines, but we never got one. A Russian team, however, has gotten some parthenote stem cell lines. We stopped the research at this point in time and are in the process of, all right, so here's the results to date. The efficiency is too low. You need 20% of eggs to give a stem cell line, or it's not worth a woman going through an egg collection, and we can go through those numbers if you're interested. We think that you need more robust early embryo development, that inner cell mass I showed you was not doing very well. And for that reason, we've put the work on hold, and we've actually now started a project with Athens in which we're looking at what is the gene expression in normal human embryos at the 8-cell stage. Once we understand what genes need to be turned on to give you a robust inner cell mass, then we'll go back to the parthenote work. This is work that's ongoing. We started our analysis with cell cycle genes. There have been a couple of papers published. This is 2009, 2010. We're about to bring out a couple more papers this year. Now let's go back to the stem cell debate. So that's just to let you know that the parthenote work I think is really valuable, and we're going to turn our energies all the way back to that as soon as we have some answers to some basic questions about what should those activated eggs be doing at the 8-cell stage. Now, whoops. So I don't think there's any disagreement about the value of regenerative medicine. The debate centers on the source of the stem cells. And there's also a debate about whether or not adult stem cells can fill the need. And I think this is an important and ongoing debate, because we may be able to do this. In the meantime, the gold standard for this is going to be embryonic stem cells. Why is federal funding so restricted? It begins in the early 80s. Scientists failed to have a constructive dialogue about IVF. In England, they had a rancorous debate about this. We didn't have one here. And consequently, in 1980s, an ethics advisory board was convened, and it was recommended that federally funded research be done on fertilized human eggs to make this new technology of IVF safe for mothers and babies. But that such research need to be reviewed by an ethics advisory board. Throughout the 1980s and the early 90s, no ethics advisory board was convened. So scientists could write up, and there are several scientists did, you could write up a grant application, it could be reviewed by study section at NIH, but it could not be funded because there was no ethics advisory board. Therefore, they could be reviewed but not funded. And what did scientists do? We sat at home and complained, but we did not march on Washington. In 1993, Bill Clinton decided to go through an NIH revitalization act. He convened a human embryo research panel, and this panel met for a couple of years. And they recommended, again, that some federal dollars be released to study fertilized human eggs under a very carefully designed system. But Clinton, again, was persuaded to not adopt the panel's recommendations. So throughout the Clinton administration, there was no federal funding available for fertilized human eggs. But during that time, things got worse, in fact. So as part of the Revitalization Act, they simply erased from the guidelines that you needed this review panel, that the Ethics Advisory Board requirement was gone. And I wrote a law review on this a few years ago, and if you look at the Federal Register the year before and the year after, it's as if that sentence had never been there. It's really kind of spooky. And an NIH administrator circulated the good news in an email to a bunch of people who'd been trying to get their work funded. And that email was intercepted by Congress. And they said, wait, you're not going to do this. You're not going to slip this in. And that was the birth of what's called the Dickey-Wicker Amendment. So in 1996, an amendment was made. It's a budget. It's a rider on the National Institutes of Health budget. And it says none of the funds available to the National Institutes of Health can be made available for the creation of a human embryo or embryos for research purposes, or research in which a human embryo or embryos are destroyed, discarded, or knowingly subject. Further, it defines what an embryo is. And included in that definition is parthenogenesis, cloning, any other means from human gametes. So this now is called the Dickey Amendment because Wicker is now a senator in the House of Representatives, and this is now renewed every single year on the NIH's budget. So when Mr. Obama became president, what he was able to do was to, so one of the things I think it's important to remember is that when Mr. Bush made the statement that he was the first president to allow federal funding of embryonic stem cell research, he was correct. Nobody prior to him had ever allowed federal funding for any human embryo or embryo derivative. When Mr. Obama became president, he lifted some of the restrictions that Mr. Bush had put in place. Mr. Bush limited the funding to what he thought was about 20 or 60 cell lines. The numbers get confused. And there were really only a few cell lines that were eligible. Mr. Obama wanted to lift that so that all embryonic stem cell lines that were available could be researched. But they put in place, the National Institutes of Health put in place such dire restrictions on the consent forms that many of the lines in existence can't be grandfathered into this because they were derived before consent forms talked about stem cells. So patients could donate their embryos for research, but the NIH wants that research to include the derivation of long-term stem cell lines. And if you donated your embryos 15 years ago, that wasn't part of the consent form. So many lines that are now in existence are not federally funded. But included in the NIH guidelines now is that no federal funding is going to be used for parthenogenic stem cells. And we don't, the only reason we can understand this is that nobody wants, the NIH does not want to get into the debate of whether it's okay for women to donate their eggs. It's not a debate they want to have because I think they're afraid that they will lose the funding altogether. So now, by a quirk, the restrictions on stem cell lines derived from unfertilized human eggs, which cannot give rise to fetuses, is entirely restricted. You can't research the lines that are derived even by private funds, and you can't derive them. It's very curious. So this is a summary slide. This is what I think effective stem cell funding should have. It needs rigorous ethical oversight, frequent rounds of funding. The field changes very rapidly. Patient advocates have to be part of this process. The ongoing, what I've learned, I sit on a couple of committees for California, and the patient advocate input into what's going on in California is huge and very helpful. You have to encourage collaboration between investigators and between among institutions. And somehow somebody's got to create mechanisms to protect intellectual property developed with private monies and still releasing that information to the public. Now, there is an opportunity now for biotech companies because excellent scientists all over the U.S. are underfunded. There's an opportunity for cost-effective collaboration with educational institutions and private industry. I think an example of this was during the stimulus funding last April of 2010, the NIH put out a one-time request for applications, and they expected a response of about 1,500 to 2,000 grants, and they got 20,000. So that is, I think, a measure of the pent-up funding needs for really well-trained scientists all over the country. When the National Institutes of Health launched the War on Cancer in the 60s, the idea was to develop extremely well-trained clinician scientists, and they have succeeded in doing that. So now we have U.S. scientists are the best trained scientists in the world, but we don't now have enough money to support them by the NIH funds. I don't think our NIH money is going to grow the order of magnitude it needs to grow in the next 10 or 20 years to take full advantage of the scientific manpower that we have. So somehow we're going to have to come up with another way to do this besides this huge overdependence on federal funds which has been developed in the last 20 or 30 years. I'll just quit here with a thank you to the board that keeps the foundation going and keeps me out of jail so far. And I'd be happy to answer any questions.
Thank you so much. We have some time. We have a few minutes for questions and comments from the audience. We have any hands? And if we could have the lights up forward. Yes, and please identify yourself.
Hi, my name is Eric Hewlett. I'm the medical school faculty. So can you explain a little more about the nature of this is a initially publicly funded foundation that is established in the state of Massachusetts and has an endowment or ongoing support from any sources? What's the nature of that?
No, the trustees funded, it's privately funded. Somebody asked me to add up the hours that I spent trying to raise money for the foundation in 2010 versus how many hours I spent writing grants, and it's about the same. No, it's just public funding initiatives. Amazingly, I mean, we have people that we obviously don't know fund us, so.
Michael Swanberg from the School of Nursing. I'm interested why women were required to have previous children prior to donating eggs for research.
There were two reasons. One is we didn't want to run into the issue that if this woman had an infertility issue later in her life that she was going to blame it on that. And the other is that the only real danger that we know of to women who are going to go through an egg collection is the hormone stimulation that she must do in order to recover more than one egg at a time. If the woman had been through pregnancy and she did not have postpartum depression, there was the thinking that she could probably handle the hormone stimulation more without as many side effects as if it was a totally unknown.
Hi, I'm Danny Becker. I'm a physician, and thank you for boiling down the molecular biology without losing the profundity. What's happening in England and Europe with stem cell IVF programs?
The English have been organized about this for some time because they had a rancorous debate when IVF first started. So their stem cell programs and their embryo work and all of that is going through the same sort of, they have a licensing authority. I can't remember the exact title of it, but it's a panel of experts and scientists. They review proposals, they approve them, and if they've got the right kind of ethical bent. And they allow this work to sort of slowly go forward. Places like Germany don't allow it at all. So Europe is a really mixed bag in terms of what they will allow. Italy, you can't even touch a human egg. It's thanks to the Pope that we learned how to cryopreserve human eggs, because the Pope decided maybe 10 years ago, that the Vatican came out with a letter that said every egg you fertilize you must return. There will be no embryo storage, there will be no embryo wastage, there will be no embryo anything. So the Italian scientists immediately learned how to cryopreserve eggs. So Europe is not a one-size-fits-all. Israel, the work is going forward very fast. Sweden, other places like that, it's going forward.
Other question? Comments?
I think everyone is aware that there's currently only one FDA-approved trial ongoing. It's the Geron trial for spinal cord injury. And that is ongoing, and it's started, I think they've done 3 patients now. It's a very defined protocol. It has to be an acute spinal cord injury in the thoracic region, which is not as common as the cervical region. And they're hoping to do 10 or 12 patients and show safety.
The source of cells?
Is one of the original cell lines derived in Wisconsin by Jamie Thompson, H9.
I understand the history of it, right? You touched on it briefly about the progress of IVF, speaking mostly about Dr. Edwards' work in England. The outcome there was an obvious positive outcome, healthy babies being born by IVF. And so I guess getting to what you just mentioned, do you think that it'll be enough for the success in the Geron trial and trials like that, real profound success that's public, to get things changed at the federal government level? Will it be enough?
I truly have come to the decision that the federal government can't fund all of this work. The federal government has to wage war, it has to fund education. It simply can't be the source of the funds that are needed to optimize the work. I think a really interesting example of that was I looked into the kind of the history, and when polio was recognized and when President Roosevelt wanted to fight polio, we had an NIH. But that's not how he raised the money to fight polio. He created the National Paralysis Foundation. So I think it's got to come, we have this over-dependence on federal funds that's grown up, that Dr. Hinton and I have grown up with. I mean, and it's to the point, if you don't have an NIH grant, you don't get promoted, you don't even get hired. And that has created an imbalance in the responsibility for the work. And so that balance has to come back to something else. And I don't know if it's increased tax incentives for donations to your favorite charity, like the foundation, if it is tax incentives to corporations to help fund basic research at universities. It's probably going to be a whole combination of things. But what absolutely has to happen is we have to get out of the mindset that it's all coming from NIH. Because, they're kind of their own big bureaucracy now. And they underestimated by a power of 10 the numbers of people who wanted to be funded under their stimulus. So that whole paradigm has to change. Maybe you don't agree with that.
No, I agree. I think you're spot on. And Eric's sure as well. And I think we've become highly dependent on funding,
Right.
and you're exactly right.
And institutions have become dependent on the indirect costs.
Absolutely.
So we've built whole empires on NIH indirect cost money, and it isn't there anymore, and it won't be there for a couple of decades. It isn't to say it won't be there again, but it's not going to be there for the next 20 years.
I think this is a good place to stop. This is a hallmark of a good medical center hour when we've just introduced a topic that could keep us here for another couple of hours. But I would like to thank Ann Kiessling for coming and sharing with us not only her scientific expertise, but her passion for the work that she does. As I listened to you, I felt like she's only getting started after all these years of work and waiting for certain other discoveries to be made. But I think you're a marvelous first award winner for the School of Nursing, and you set a very high standard for the subsequent year's awards for everything that you've brought to us today, and thank you so much. We'll hope that you join us next week for the last Medical Center Hour of the season. It's a program called Asylum, Inside the Closed World of State Mental Institutions. It's a photographic exhibit. The architect, photographer Christopher Payne will be with us. The exhibit is already up in the School of Architecture's Almala Gallery. You can see it weekdays between 8:30 and 5. Your handout has some information about other events also next Wednesday in association with the exhibit. But we hope you'll join us for this, which is also our closing partnership, the Crispell Lecture, with the History of the Health Sciences Lecture Series. So we'll see you then, and again, thank you to Ann Kiessling and to the School of Nursing.